Hello students, welcome back to the biology class. So in the last class we covered the chapter number 8, that is the human health and diseases. So we have covered that complete unit also. Now today we are going to start with a new unit in which the first chapter is regarding the biotechnology principles and processes. There are two chapters to cover in this unit, followed by two chapters of ecology, and then your syllabus would be covered. So the topic of biotechnology is it's regarding how the technology of the microscopic world, if they can be used in the living organism or inside a cell, inside the tissue, or maybe inside a living complete macroorganism, or otherwise the microorganism. So the techniques, if they can be used into a living organism, then we can start preparing things like that. Means these microorganism or macroorganism would become something like a factory for us to synthesize products of requirement or product of daily use. So this concept is called as biotechnology. We all associate the word technology with the artificial world, the world in which machines are there, or there are setups or equipments or laboratories. But what is biotechnology? Using similar type of concept but in the living organism. So the living organism will work like a living manufacturing factory for us, synthesizing products which we require or making business out of this. So biotechnology is the technique of using living microorganism or plants or animal cells or their components or enzymes from the organisms to produce products and processes to serve to the human beings. So the biotechnology word was coined in 1917 by the Hungarian engineer Karl Erke, and the father of genetic engineering is Paul Berg. Now if you read the NCERT, you won't find the name of Paul Berg anywhere much mentioned. But yes, there is mention of the Herbert Boyer and Stanley Cohen, which we will be talking in the today's class. So the recombinant technology, it is biotechnology is also called today as recombinant technology. Now what exactly is recombinant? When you combine the two different sources of DNA together to form a new DNA which is a hybrid of both, you call it as a recombinant DNA. And this technology is called as the recombinant DNA technology, also mentioned as small r DNA technology. So the whole concept of this principle of biotechnology deals with the techniques of using living organism or enzymes from these organisms to produce products, and then these products are given to human being for their utility. In this sense, making curd, bread, wine, remember all these things are also a part of biotechnology. So if you go through NCERT, you will find there are mention of two types of biotechnology: old type of biotechnology and the modern type of biotechnology. So when we make bread, when we make alcohol, when we make yogurt, any type of South Indian food, all those things are using technology in the living world. But that technology was not so much sophisticated or complex. That technology only tried to exploit the capabilities of living organism, the natural capabilities. So in the old or traditional biotechnology, the natural capabilities of organisms or cells are exploited, and then we start preparing from them alcohol, yogurt, etc. And remember, even when we talk about plant breeding and animal breeding, even those concepts are also a part of old or traditional biotechnology, because in this concept we are simply using two animals or two plants and consider them as male and female, and then we crossbreed them. So when we are crossbreeding two plants or two animals of our desire, we are only exploiting their natural capabilities. Like, it is not something like a new type of expression will be there. It's only the expression of those parents which will come up. So that's why this type of technology has has a shortage, because the problem is that we cannot bring something new into the characters of the organism. So it only exploits the natural capability of the organism. So when we prepare these products of daily use, these are microbe mediated processes, and these are also a part of biotechnology. But then came the concept of modern technology of biotechnology, that is the genetic engineering. Now the production of the techniques which are based on the manipulation of the genetic material. So when the genes or the DNA of an organism is modified, now this modification became possible because by the time plasmid was under study, there was a scientist, Stanley Cohen, who was performing a lot of experiments on plasmid, how to remove plasmid from bacteria and how to insert into another bacteria. Such type of experiments he was interested in. And then came the concept of restriction enzyme. We'll be learning in detail about all these things. So the restriction enzymes are the enzymes which are today called as the molecular scissor, also called as biological scissor, and these scissors were able to cut DNA precisely at a particular location. So these enzymes cannot cut the DNA anywhere randomly, but they cut at a particular place, and if that place is repeated 10 times, then 10 times cut would be made on the piece of DNA. So these type of discoveries made it possible to manipulate the genes or manipulate the DNA of an organism and to prepare a new type of DNA, which will be the recombinant DNA.